The overall goals of this procedure are to analyze the metabolic activity of a biological sample and to determine the extent that anaerobic pathways contribute to the cellular energy production within the sample.
This method can help answer key questions in the pharmaceutical field about the extent of mitochondrial toxicity during drug development. The main advantage of this technique is that it facilitates the simultaneous measurement of oxygen consumption and heat production in a cell sample of interest.
So this method can provide insight into treatment planning during pharmaceutical development. It can also be applied to other fields, such as cryobiology, in which the effects of metabolic preconditioning on cryopreservation can be assessed.
To prepare the respirometer, first add 2.2 milliliters of DMEM to each respirometer chamber and fully insert the stoppers. Adjust the stoppers with a stopper spacer tool to allow optimal oxygen diffusion to the medium, and set the chambers to continuously stir the samples at 750 rpm at 37 degrees Celsius until the oxygen concentration and the oxygen flux are stable. Confirm that the respirometer software is programmed to account for the oxygen solubility of the medium used in the experiment. Then, select a stable portion of the oxygen concentration trace and calibrate this portion for 100% air saturation. Now close the stoppers, taking care that no bubbles are present in the chambers. After about 10 minutes, a stable oxygen flux should be observed. To prepare for calorimetry, first add